obesity. So today I will first define childhood obesity, what's an obese child and how are we going to define it. Then I will talk about screening tools which we can use and which are hot topics now in research of childhood obesity. First, the single nucleotide polymorphisms and gut microbiota also. Then I will talk about obesity and related metabolic dysfunction or metabolic disorders in childhood obesity. And at the last part, I will talk about screening parameters which we in the future can use to detect metabolic disorders in obese children. So first, how to define obesity? Obesity is a state of an increased body mass or increased body weight. It's due to the fat accumulation, so the accumulation of adipose tissue, which can eventually produce adverse health effects like diabetes, cardiovascular disease, but also some cancers. And to define an obese child, we have to measure. We have to measure first the weight and height to define the BMI. We calculate the BMI as being weight over height squared. In an adult, we see adult with normal weight has a BMI between 19 and 25. An adult who is overweight has a BMI above 25. An adult who is obese has a BMI above 30. But we can also have a certain class of obese, so a BMI above 30 is class 1, BMI above 35 is class 2, and morbidly obese adults are identified with a BMI higher than 40. But in children, it's different. Children grow, and their body size increases during the growth process. So we have to account the age, but also gender. Because we see in girls, they have a higher amount of fat percentage of body mass. So we have to calculate this and take this into account. So we have to adjust the BMI. How do we do it? We have, or they developed, researchers developed BMI centiles. And in Flandria, we use the Flemish cutoff points. In clinical practice, we use the growth charts for BMI. And I don't know if you see it that well, but there are gray zones. And the upper gray zone is in which children are defined with overweight, and children with underweight are in the lower gray zone. Children with normal weight are presenting in the white zone. So we have, like centiles, it's the BMI in the y-axis and the ages in the x-axis. And we see, for example, we have a 14-year-old boy with a height of 175, a weight of 89. And we calculate the BMI, so we see BMI is 29. And we see he is 14 years, so he is defined as being overweight according to the Flemish cutoff criteria. If a child is obese, he's above the upper gray zone. But in research, we have to use the international criteria. So the same example will be obese according to the international criteria. So for some people, some children, there is a difference in Flemish criteria and international criteria. So defined as overweight in Flemish criteria, defined as obese according to the international cutoff points. But in research, we use the international, and in the clinical practice, we use Flemish criteria. We also can calculate the BMI standard deviation score or BMI set score. It's with a formula, and we calculate it with a software, some program, which does it for us. Then a child with overweight has a BMI higher than 1.3. A child who is obese has a BMI higher than 2.3. The BMI calculation is simple, it's quick, it's also inexpensive, but the problem is we cannot discriminate between fat and muscle mass. So we see, like, the fitness boy has a high muscle mass, an obese boy has a high fat mass. If we calculate the BMI, they have the same BMI, but according to the fitness boy is more healthier than the obese boy. So we also can measure body fat. distribution. We do it with waist circumference, so we measure the waist, or we measure the hip. And that's a parameter of abdominal obesity, which is a high risk factor for diabetes or cardiovascular diseases. For the waist circumference, we have Dutch references cut off for boys and girls according to age, so the cutoff criteria exists. We can also use waist to height ratio or waist to hip ratio in research. And for mid-upper arm circumference and neck circumference, we measure it less, but it can also be measured, and mid-upper arm circumference is a measurement of central obesity, and neck circumference of upper fat distribution, but they are not commonly used, and also there are no cut-off criteria for hip circumference, mid-upper arm, and neck circumference in children. But it's a quick and simple tool, so we use it also. Then we have the skinfold measurements, which are measured on 4 regions on the body, so biceps, triceps, subscapular, and abdominal. And we measure it with a caliper. And so it's like squeezing the skin and measure the millimeters on the caliper device. Then there can be used some equations to calculate some of the 4 skin folds and to define the percentage of body fat. But it's a difficult device to use because children don't like it. You have to squeeze the skin and it hurts. But it's useful in longitudinal studies, so if we measure it now and we measure it within a couple of months, we can see if the body fat is increased. We also have national cutoff values, but in research it's not that reliable because we have inter and intra-observer variation. If I measure it or the doctor measures it, there is a variation, so it has to be always the same person who measures it. It's also time expensive to measure it, and we have to measure it in 3 times to be exact. In extreme obese individuals, it's also difficult because if you want to squeeze the skin, it's not that easy when there's too much fat on the body. But we also need different equations to calculate eventually the sum of the 4 skin folds. So at the moment I'm using bioelectrical impedance analysis, which is a quick, simple tool. It's also a portable device. It works with an electrical current throughout the body. So we have to put some stickers on the hand and the feet and then connect the electrodes with it, and then there is a current throughout the body, about 5 to 50 kilohertz, but the children don't feel it. It's not like we electrocuted. It's based on the resistance of the cell membranes and the extra and intracellular fluid. So it measures the whole body, it measures the hydration, so the cell membranes, extra and intracellular fluid resistance, and eventually so it can calculate the overall existing body fat. It's non-invasive, it's also safe, and it's reproducible. So if we measure it now at the moment and we measure it within a week, it will be the same if the fat percentage did not increase, but it's very reproducible. It's less accurate than the DEXA. DEXA I will talk about later. And the problem is also if they have previous exercise, they are more dehydrated, so the hydration level will be different and the fat percentage could be overestimated. So we also could use densitometry, but in children it's difficult because it's scary for them to, it's like underwater weighting. It's scary and not practical. It's also expensive, but it's an accurate method which is based on the measurement of the body density. It's based on a 2-compartment model of fat and the fat-free mass. We could also use the BOD-POD, which is like the same approach, but it measures the airflow and not the water. But it stays in one place, so we have to. It's not practical, it's not portable, so it's not used in our clinical practice. Then we also have the DEXA scan. DEXA scan is originally used to measure the bone tissue, but can also detect muscle tissue, fat tissue. It's easy and it's also quick. It's like 10 minutes. The person has to lay down under a scanner. But the problem in children it has radiation, so it's not ethical proof to put children under the scanner. There are standards being established, but it's also expensive, so we don't use it. And it's limited availability in the hospital. There are much persons who has to be under the DEXA scan, so it's not always available. So how big is the problem? We see overall the world, we see that especially in the United States, we see 35% of the children are overweight and obese. And also in Canada, but also in Brazil or Australia, the percentage is high. And we don't see only in developed countries, we see the increase in obesity, but also in developing countries, the obesity problem is raising, because also it's like a status. If you are overweight, you have how do you say it? You have much money to eat and you have good status. So in developing countries, it's also increasing. In Belgium, it's like 14-15% of the children are overweight. Of about 5% are obese. So it's not that big problem, but we tend to follow the US. So we have to take into account that will become a bigger problem. So if you calculate, it's 1 out of 25 children in Belgium who are obese, so 1 out of a class of students of children will be obese, for example. There was also research about the estimation of the trend, so that we see in 2020, preschool children will be, so the increase in obesity will increase until 2020. Global increase. In Belgium, we see stabilization of obesity, but this does not mean that we do not have to prevent it. But the global prevalence is increasing and will be increasing. So the problem of obese children is that they are at higher risk for a couple of comorbidities. First of all, we have the fat mass, which stresses on the body, on the bones, and it can increase the mechanical load, eventually leading to orthopedic problems, so problems of the knees, of the hip, and so on. Also, if children sleep and they lay down, the fat pressures on the body, and they get respiratory problems, and they stop breathing during their sleep. Also, there are psychosocial concerns related to it. So children who are more fat than other children, they have like psychological problems or lower self-esteem. But also if they have a sleep apnea, they can have concentration problems in school, learning difficulties, and eventually they have a poor quality of life. They can also develop some forms of cancer, like bowel cancer, pancreas cancer, liver cancer. And the main concern is the increasing fat mass. I will be talking about or focus on this part, because fat mass can excrete some like cytokines and hormones, eventually leading to insulin resistance, hypertension, dyslipidemia, or a low-grade inflammation, or a fatty liver. And this can lead to the development of metabolic syndrome, diabetes, or cardiovascular diseases. Also, children which are obese are at higher risk to early develop during puberty. So girls can have the PCOS syndrome, and boys they can develop hypogonadism. And eventually, they are at high risk to have an impaired fertility. But the main concern is that 70% of obese children are also obese in adulthood and eventually also developing diseases like diabetes, cardiovascular diseases. So we have to prevent obesity, then we can also prevent the development of other diseases, cancer, diabetes, and cardiovascular problems. So what's the problem? What's the cause of childhood obesity? In general, it's due to an energy imbalance. So there are too much calories consumed compared to the calories expended. Mostly it's due to an unhealthy lifestyle, the bad nutrition, too much food, too much bad food, too much fat, too much carbohydrates, compared to a low level of exercise, so a low physical activity. Also, psychological factors can be a cause, like divorce of the parents or some other problems. Social status, so the education of the parents. Parents also have a very high impact of the development of obesity of the child. There are also parents who don't recognize that their child is obese. That's also a problem. Genetic factors also can contribute to the development of obesity. There are rare Mendelian disorders like the Prader-Willi syndrome, but are also rare monogenetics, except for the MC4 receptor. That's a mutation which is more commonly seen in obese children. We also have the common polygenetics, like the single nucleotide polymorphisms, which are little variations in the genetics and is recognized to be important in obesity. Other predisposing factors are prescribed drugs like antipsychotics, or drugs that can develop weight gain. Also, the toxic environment or gut microbiota could be important in the development of obesity in childhood. So you see, it's a multifactorial and complex disease. So now I will be focusing on the single nucleotide polymorphisms and the gut microbiota. So they can be used as screening factors of obesity. First, genetics of common obesity. 40 to 80% of the variation and the increase in BMI or also the obesity risk is due to genetic factors. In genetics, we have two approaches we can use in research. So we have the genome-wide association approach. It's more like a hypothesis generating approach. So we focus on a whole population, and then we detect a couple of or a lot of genetic loci which can be important in obesity. And already they found that more than 15 genetic loci are associated with obesity, but less than 2% is explains the inter-individual variation in BMI. So we have to do a lot of other research. We also have the candidate gene approach, it's hypothesis-driven approach. So we going to focus on candidate genes which could be important in the obesity process, and eventually going to search in a population, obese, not obese, and see whether the candidate gene is important in the obesity process. Till now, there are 127 candidate genes associated with obesity traits, and 12 genes are established already in more than 10 studies. So single nucleotide polymorphisms, what is it exactly? We have a variation, for example, in the genes between persons. Persons can be homozygous for example, a T allele. They can be heterozygous for the AT, and then can be homozygous and have an AA allele. Then there can be like an error in the transcription process, or during your lifetime it can change, and eventually it can discriminate people with a change of base pair on a locus, on an allele. So we have one SNP per 300 base pairs, which means that we have 10 million SNPs into a human body. We see, for example, if we relate it with the BMI, we can see that, for example, some people which are homozygous for the A allele have a higher risk to increase in BMI. in a certain gene, for example, leptin. And we see that persons with homozygous for the T allele have less risk to increase in BMI. So the T allele could be, in this example, could be protective against the increase of the BMI. So you have to take into account that SNPs don't have, one SNP don't have the whole, does not act, but it are all SNPs together, so we have to focus on a couple of SNPs and not only at one SNP. You see they have a cumulative effect, so they work together to increase BMI, for example, sum of skin folds or waist circumference. So you have to take the whole package of different kinds of SNPs together. If we see a recent study already proved that a SNP in the FTO gene, a gene important in obesity, that subjects with homozygous for the A allele had a higher increase in BMI compared to subjects homozygous for the T allele. And then also they saw that a SNP in the MC4 receptor gene had a higher, subjects with a homozygous for the A allele had a higher triceps skin fold compared to subjects with the G homozygous for the G allele. So another study searched for more SNPs, and they found that especially MC4, the FTO, but also TMEM and BDNF are important in the obesity. So to give you an example, if there is a SNP, for example, in the MC4 receptor or BDNF, it dysregulates the energy, or could have an effect on the dysregulation of the energy balance. So here we see the leptin signaling pathway. It's complex, but I'm not going to explain the whole figure. It's just to give you an idea. If adipocytes increase, leptin will be increasing and have an effect, a positive effect on receptors in the hypothalamus, but also a negative effect. And eventually, through signaling pathways, it will activate or deactivate MC4 receptors or milk-protein receptors, which again have an effect on BDNF. So if here is a problem, eventually here will also be a problem. So less food intake will become more food intake and less energy expenditure. So now I will talk about gut microbiota. It is a hot topic at the moment in research of childhood obesity. Because we have the cause of childhood obesity, genetics, environment, but the gut microbiota is also recognized as contributing to the chronic positive energy balance in obesity. So we see gut microbiota, what is it? It's our bacteria living in our gut, living in the mucosa or in the lumen. There are trillions of bacteria, and there are 100,000 times more bacteria on your body than there are humans on Earth. You have also 10 times more bacterial cells than human ones, and you have 100 times more bacterial genes than human ones. So we are more bacterial than human, if you see so. There are 500 to 1,000 bacterial species, and we see in the stomach or in the duodenum there are less presence of bacteria. The highest presence is in the colon. So we focus on the colon, and we can detect gut microbiota composition through the feces. So we detect the presence of microbiota in the feces in research because it's difficult to see the gut bacteria composition in the colon. There are 4 major groups. Firmicutes, Bacteroides, Proteobacteria, and Actinobacteria are the main phyla of bacteria. Your flora is your friend. Why? They perform a couple of functions for us in the body. So food that we cannot digest by ourselves, they are digesting it. So they ferment non-digestible polysaccharides or dietary residues. They also help to protect against pathogens, and they provide essential nutrients like vitamins, vitamin K, vitamin B, and they help in the development of the immune system. So during our lifetime, we develop this unique gut microbiota. First, the fetus has a sterile gut microbiota. Gut means that there are no gut microbiota into the gut of a fetus. When the baby is born, during birth and after birth, it gets the bacteria from mother and also from the environment. After 1 week, there is a habitation of Enterobacteria and Streptococci into the gut of the baby. After 1 week, we see that Bacteroides, Bifidobacterium, and Clostridium are residing in the gut of the infant. And after 2 years of age, we see a stabilization of these bacteria. So before it's unstable. And for example, one day Bacteroides is the highest, and the other day Clostridium. So at 2 years of age, it's stabilized, and at 4 years of age, it's fully matured. So you have a good microbiota composition of your own, and unique. But there are also overlaps in gut microbiota composition between individuals, meaning that 1/3 of the species are present in almost all individuals. So it's like Obama and Romney, they are competing, but they also have a substantial similarities, so they share 1/3 of their gut microbiota. That means 38% of the gene pool is shared by at least 50% of the individuals. So what's the link between gut microbiota and obesity? So the hypothesis is that in an obese subject, there is a different composition of gut microbiota, leading to a higher energy extraction out of the non-digestible food components. So certain species of bacteria have a better capacity to extract energy out of the food. There are also bacteria maybe who produce inflammation, LPS, or LPS also blocks the production of, or the action of fasting-induced adipose factor, and eventually this can lead to inflammation of the liver, adipose tissue, hypothalamus, or muscle. It can also lead to a lower insulin sensitivity, so insulin resistance in the organs, or macrophage infiltration, so inflammation again, and an increase of lipoprotein lipase. So in animal studies, they already saw that an obese mice, leptin-deficient obese mice, had less Bacteroides and more firmicutes compared to the lean mouse. And they also found that they had an enrichment in genes important in the breakdown of those indigestible dietary polysaccharides. Also, they have an enrichment in genes that encode enzymes important in the metabolism of fatty acids, but also sucrose, meaning that this composition of gut microbiota have a higher capacity to harvest energy from the diet in obese mouse. So there's also an experiment which compared mouse with a low-fat, high polysaccharide diet compared to mouse which they gave a Western diet. And they saw that the mice with a Western diet had more molecules. Molecules are a bloom of the firmicutes. And the mouse with the low-fat diet had more Bacteroides. So there is also seen a dramatic fall in diversity, meaning that different species are less present. Also, there were alterations of the metabolic potential in mouse consuming the Western diet, so there are enrichments in genes encoding in the energy metabolism, and the consequences were that the obese mouse had an increased capacity to import these carbohydrates from the Western diet. And they have also increased capacity to metabolize the sugars into short-chain fatty acids. In human, they also saw that there was a reduced diversity. They also saw that obese subjects had higher firmicutes, lower Bacteroides, and in subjects with weight loss, they also saw a decreasing in firmicutes, so they saw the same. And also altered representation of metabolic pathways, so they saw a decrease in the butyrate concentrations. In obese children, there are also some studies performed. We see that in infants, an increase in Bacteroides fragilis group was related to an increase in the BMI SDS. The development of overweight, so children which developed overweight at the age of 7, at the age of 1 month, they had lower bifidobacterium and higher Staphylococcus aureus. Also, some other bacteria could be important and were up or down regulated into the gut of child obese children. And there was the last study, 2012, it's not a long time ago, they saw that in obese children they had higher prevalence of vermicutes, and in children had a higher prevalence of Bacteroidetes compared to the obese children. So the aim of our study was to see which gut microbiota species were predominant in obese children and compare it between children, and also the effect of diet and physical activity. We wanted to see if there is an effect of diet and physical activity on the gut microbiota, which eventually can contribute to obesity. So we included 26 obese children, 27 non-obese children from 6 to 16 years old. We asked them to fill in a food diary, a physical activity questionnaire, and also to give a stool sample to detect the gut microbiota species in the feces. And we selected 5 predominant fecal bacteria. We selected Bacteroides, Bifidobacterium, Clostridium, Staphylococcus, and Lactobacillus. And we detected the presence of these species with quantitative plating on one hand and PCR on the other hand. Quantitative plating is the gold standard technique, so we culture bacteria, so we let them grow on media, on specific media, and then we count the number of bacteria present on the media, and we express it in log cells per gram feces. And the PCR procedure, it's more accurate, it's also gives more results. We focus on the 16S ribosomal region of all bacterial sequences, and we use primes and probes to detect a specific bacterial species, and eventually also it was expressed in log cells per gram feces. Additional to quantitative plating, we use MELDI-TOF-MS, which is a technique in which the protein structure of a specific bacterial species is detected. So we focus on the identification of Bacteroides fragilis. So this is the medium for Bacteroides. And then we detected Bacteroides fragilis group subspecies and expressed it in percentage. So this method is easy, and eventually a database is inserted into the device, and it gives us a certain species and a score value of the presence of the species in the sample. So in general, we saw logically that the BMI SDS was elevated in obese children. And we also saw that, also logically, that protein expressed in gram per kilogram body weight was different between obese and non-obese children. But according to the guidelines, we have to use protein gram per kilogram body weight, but there is also a discussion on it because in obese children it gives a wrong idea of protein intake, because we have to divide between kilogram body weight. So obese children have a higher body weight, so have a lower intake of protein. So we focused on the energy percentage of protein, and then we didn't see any difference between the intake. When we looked at gut microbiota and quantitative plating, we saw that there was borderline significance between non-obese and obese children, meaning that non-obese children had a higher percentage of Bacteroides fragilis compared to obese children. For the other, we didn't saw any significant differences. And the additional MELDI-TOF experiment shows us that Bacteroides vulgatus, so subspecies of the Bacteroides fragilis group, was highly expressed in non-obese children. We see also that Bacteroides vulgatus is like a good bacteria for our gut. So this kind of explains why it's highly expressed in non-obese children. For the others, we also didn't saw any significant difference. For the PCR technique, we detected a higher level of lactobacilli in obese children, so lactobacilli could also be important in obesity. When we calculated the ratio of firmicutes to bacteroidetes, it's according to an article, we detected the ratio and we saw that the ratio was higher in obese children, meaning they had higher percentage of firmicutes compared to bacteroidetes, and non-obese children, it was the other way around. So this was highly significant. When we looked at the diet, associations of diet, physical activity, and gut microbiota, we saw that carbohydrates and energy percentage was positively related to Bacteroides fragilis group, detected by quantitative plating. Energy and calories per day was negatively associated with the presence of Parabacteroides dehiscessonis, which is a subspecies of the Bacteroides. When we looked at the bacteria detected by quantitative PCR, we saw that fat expressed in energy percentage was positively correlated with Bacteroides, protein was negatively correlated with Staphylococcus, and physical activity level was positively correlated with Clostridium leptum. We also did some confirmation, so we did correlation analysis to see if some factors were positively correlated with the BMI SES, so which bacteria were more or less pronounced in obese children. But it's the same with which I talked about earlier. In conclusion, we saw significant associations between diet, physical activity, and certain gut microbiota species. We saw that there was a higher carbohydrate intake was related to the presence of more Bacteroides fragilis. A lower protein intake was correlated with more Staphylococcus species. A higher fat intake was correlated with higher Bacteroides in the gut. A higher energy intake was correlated with lower Parabacteroides dehiscessonis, and a higher physical activity level was correlated with the presence of Clostridium leptum. So eventually there were alterations seen between obese and non-obese children. So obese children had a lower level of Bacteroides fragilis, including a higher level of Bacteroides fragilis subspecies and a lower level of Bacteroides vulgatus. We also saw that there was a high presence of lactobacilli, and the ratio for Firmicutes bacteroidetes was elevated. So lactobacillus and also subspecies of the Bacteroides fragilis group could be important in the causes of obesity. So if we reveal the gut microbiota composition in childhood, it may lead to develop early screening tool to identify obese prone children. And also microbial manipulation through diet, antibiotics, probiotics, prebiotics could manipulate the gut microbiota and an early life stage, for example, and prevent the development of obesity and related disorders. So now I'm going to talk about obesity and relation with metabolic dysfunction. So we see in some children, but not in all, they develop, the same time with obesity, they develop also insulin resistance in the body. And they have a higher risk to develop metabolic syndrome and diabetes. And when they develop metabolic syndrome, they have 2 times higher risk to develop cardiovascular disease and 4 times higher risk from diabetes to develop cardiovascular disease. So in obese children, mostly there is seen progress deterioration in metabolic function. So in the picture we see that in the hypothalamus there is a dysregulation of energy expenditure and intake, so lower expenditure, higher intake, leading to an increase in fat mass. This fat mass, it's like an endocrine organ. It secretes some important factors which eventually can have an effect on the inflammation, low-grade inflammation, and also to an increase in lipolysis, we see an increase in free fatty acids, which eventually can make the pancreas a fatty pancreas and disturb the beta cell function of the pancreas, which is important to secrete insulin. So there is going to be hyperinsulinemia, so a more secretion of insulin, leading to insulin resistance of other tissues. So if children are insulin resistant, there are also additional risk factors for a child. So if a child has visceral fat or is more allogenous or is in puberty or has a family history of diabetes, is a girl has also higher risk, or a lower birth weight, or it can also contribute to the development of insulin resistance. So not all children with obesity develop it, but there are most children develop it. And eventually there is a decrease in insulin action in muscle liver, and this can lead to a couple of complications. So first of all, I want to focus on insulin resistance. How do we detect it in obese children? So we do not use, but in research it could be used, the hyperinsulinic glycemic climb. It's a very accurate method, but we do not use it because it's time consuming and it's also difficult to perform. It's like 240 minutes, like 4 hours. It's also expensive, and we do not use it in clinical practice. But there's also, this allows the differentiation between hepatic and muscular insulin resistance. So it's an interesting tool, but in our children in the clinical practice we have to use simple surrogate measurements to screen for insulin resistance, which are derived from an oral glucose tolerance test. So an oral glucose tolerance test is, we measure fasting blood samples, glucose and insulin. Then we give a glucose bolus to the child, so it drinks like glucose. And then we're going to measure at time 30, 60, 90, 120 minutes, we're going to measure glucose and insulin, how it reacts on the glucose bolus in the body. And we can calculate some values to see if children are have hyperinsulinemia or are insulin resistant. So we can calculate the sum of, so this are the glucose values, these are the insulin values. We could calculate the sum of the insulin values throughout the oral glucose tolerance test. If it's more than 300, we can say the child has hyperinsulinemia. We can also measure fasting values and say that fasting insulin, if it's higher than 25, the child has too high insulin. We can also calculate OGTT-derived factors like the whole body insulin sensitivity, or the HOMA-IR. If it's higher than 2.68, the child is insulin resistant. So this is according to several researches and articles, but these are surrogate indices and they are not used to define children with insulin resistance or hyperinsulinemia, because there's no consensus about it, and we cannot say these are good parameters to use in clinical practice, but it is used in large studies, epidemiological studies, to see if there is an increase in insulin resistance or a decrease, but it's not used to define insulin resistance. So we have to, they have to develop indices for it. Then I'm going to focus on development of the metabolic syndrome in these children. So metabolic syndrome, also called insulin resistance syndrome, is defined with the presence of central obesity plus any two of these other risk factors. So hypertension, like an elevated systolic blood pressure, diastolic blood pressure, but also an elevated glucose, or dyslipidemia with a high triglycerides level or low HDL, so low good cholesterol levels. So the International Diabetes Federation already has some criteria developed for children. So for children from 6 to 10 years old, from 10 to 16 years old, and children older than 16, they have different cutoff points to detect if children have a too high level of glucose, too high level of triglycerides, and so on. And currently, these are the definition that's mostly used in research, but it's also a definition which has a lot of discussion on it, because these are also adult cutoffs, so for children it's not yet established. But we see, according to this definition, we see an obese pediatric population has about 15 times higher presence of metabolic syndrome compared to elite population. So I did a study on a database from a database of 2004 to 2012, and I see in 168 of these children from 10 to 16 years, I selected them according to the criteria of IDF. I saw that 36%, 36.5%, suffered from the metabolic syndrome. And I saw that in the metabolic syndrome, especially the presence of so obesity plus 2 risk factors was most pronounced. And then I saw obesity plus 3 factors and obesity plus 4 factors was less pronounced. I also saw that especially hypertension was the main factor in all children which contributed to the metabolic syndrome presence. Also, a low HDL cholesterol was highly prevalent. I also saw that 14.7% of these children had an elevated 2-hour glucose. So after the glucose tolerance test, we saw that the 2-hour glucose was higher than 140, meaning that they had like impaired glucose tolerance. And we saw that 14.7% of the children had it. So our question was now that it remains to be shown, so nobody, I've said it before, if the metabolic syndrome or impaired glucose tolerance has the highest risk to eventually develop in diabetes. So we have to know that we can look at the metabolic syndrome or at impaired glucose tolerance or at both. Also, a problem in definition of the metabolic syndrome is there are lots of definitions developed by different researchers. So we get a difference in prevalence. So according to the IDF, we saw that 36% of the obese children had metabolic syndrome. But according to the other definitions, we saw that they had higher prevalence of metabolic syndrome. So definitions has to be become universe, and that we can use the common guidelines of metabolic syndrome. So at the moment there is a lack of internationally reference values. So also dyslipidemia is an effect of obesity in a child. We can screen for it according to lipid levels. These are also developed by a researcher, and he said that the desired level of total cholesterol was lower than 150. The bad cholesterol lower than 100, HDL higher than 45, and triglycerides lower than 100. But also there's no consensus about the triglyceride levels in children. Then we can also screen for impaired glucose tolerance and diabetes. So according to the glucose tolerance tests, which I mentioned before, we see we can detect fasting levels of glucose, and if it's higher than 100, we define a child as having impaired fasting glucose. If the 2-hour glucose, so at 120 minutes, is higher than 140, 200, we said the child has an impaired glucose tolerance. And if it's higher than 200, we say the child has diabetes. So if the child has an impaired fasting glucose and or an impaired glucose tolerance, it's defined as having pre-diabetes. But there's also disagreement on it. So some researchers said we only have to focus on fasting levels, but other researchers said we have to focus on both. Other researchers said other things. So in our study sample, we saw that if we took it together, 21% suffered from pre-diabetes, but we still don't know if pre-diabetes or the metabolic syndrome is the best screening factor for diabetes eventually. So you see early detection and prevention of obesity is very important to prevent also metabolic diseases related to childhood obesity. It's also crucial for children's quality of life and the high medical care costs. So early screening for obesity is important. Early screening for insulin resistance, metabolic syndrome, prediabetes, and early cardiovascular complications. But which screening factor is most predictive and can easily be used in clinical practice? So we don't know yet, but I thought that maybe the shape of the oral glucose tolerance could be important, because nobody, there were a couple of researchers, but nobody detected in obese children and looked for it in obese children. One article related the shape, one kind of shape related to the future risk of diabetes, but it was in an adult sample, and he saw that, for example, the monophasic shape was the shape which was most predictive of diabetes. But other researchers also saw that there is a biphasic shape, which is an increase, decrease, and again an increase. And they also defined like a shape index, a simple index to use, and a simple index like BMI, for example, which you can use to easily correlate with other factors. The shape index was defined as glucose at 120 minus the glucose at 90. So this is a shape index for monophasic, which is negative. For biphasic, it was positive. But there were also researchers who found a triphasic shape, so with two peaks, and then the shape index was not defined as being glucose 120 minus 90 because it could correlate with monophasic, and triphasic they saw that was more correlated with biphasic. So they shared like the same characteristics. So triphasic was defined as glucose 90 minus glucose 60. So then they saw indeed that subjects with impaired glucose tolerance had a very high risk to develop future type 2 diabetes, and were eventually most of the subjects had a monophasic form. So monophasic was more the pattern in which impaired glucose tolerance, but also subjects with insulin resistance or lower early insulin secretion had especially a monophasic shape. And adults with, for example, biphasic shape had a better glucose tolerance, better insulin sensitivity, and better insulin secretion, and also adults with a triphasic shape had a better metabolic health, for example. So, but in obese children, we didn't know yet. So the hypothesis was that the shape harbors metabolic information, which is not captured by the level of glycemia, so not captured by prediabetes alone. So we looked for the shape in obese girls, postmenarchal obese girls. Why? Because we didn't want to include puberty, because puberty is a risk factor of insulin resistance and can induce more insulin complications. So we focused on one homogeneous group of postmenarchal obese girls. And we wanted to examine if insulin sensitivity, secretion, or the metabolic syndrome has a relation with the kind of shape of the oral glucose tolerance test. We also include girls taking no medication, because medication can indeed influence the pattern of the glucose tolerance curve we take. So we calculate the BMI. We also measure the blood pressure to eventually define the presence of metabolic syndrome. So we did an oral glucose tolerance on all these girls, and we can calculate girls with a normal glucose tolerance, impaired fasting glucose, or impaired glucose tolerance. Then we calculate a couple of indices, and for example, fasting indices like the quantitative insulin sensitivity index. We also calculated homeostatic model assessment for insulin resistance, but also for beta cell function. We calculated also indices derived from the whole OGTT, which is the insulin sensitivity index or insulinogenic index. These are all indexes which can say something about insulin sensitivity. We also calculate area under the curve for glucose and insulin, and the shape index according to earlier literature. And we defined the metabolic syndrome according to the IDF criteria. So we saw that all 72 subjects had like a pattern more monophasic, but if we discriminate them, we saw that most of the subjects had a monophasic shape. So it increased and again decreased. And we saw that seven subjects had a pre-diabetes or a disturbance in their glucose tolerance. In the biphasic shape, we saw that 27 subjects had the biphasic shape, and less, so like 5 subjects had an impaired glucose tolerance. And there were less subjects were in the triphasic shape, and none of them had impaired fasting glucose, and only one had impaired glucose tolerance. So then we compared between the two groups, like we took monophasic and BR triphasic, because BR triphasic were almost the same, had a good metabolic pattern. Then we see that the BMI SDS, the BMI standard deviation score, was eventually higher in monophasic subjects. Also, the area under the curve for glucose was higher, meaning that the glucose tolerance was poor in these children. We also saw that the good cholesterol was lower in these children with monophasic shape, and triglycerides were significantly higher. So eventually we also did a correlation analysis with the calculated shape index and a couple of parameters, and indeed we saw that the lower the shape index, the higher the BMI. So the more monophasic, the higher the BMI. Also, the higher the area under the curve for glucose. So it was like a confirmation of our tests. And we also saw that the shape index was negatively correlated with the insulin sensitivity of the beta cell, meaning that monophasic had a higher insulin sensitivity or a better beta cell function, and whole body insulin sensitivity was positively correlated with the shape index, meaning that the more B and triphasic, the better the insulin sensitivity was. So we also correlated this with the metabolic syndrome, and we saw the more parameters of the metabolic syndrome, the lower the shape index. So the more monophasic, children of monophasic pattern had more components of the metabolic syndrome, so were more metabolic disturbed, and they had a lower HDL and higher triglycerides. So eventually you can see that in these obese girls, the monophasic form was indicative of a metabolically unhealthy phenotype. So children with a higher BMI-SDS, poor insulin sensitivity, poor secretion, and impaired glucose metabolism, compared to the B and triphasic form, which children had a lower BMI-SDS, a good insulin sensitivity and secretion, and also a good glucose metabolism. So eventually these are defined as a metabolically healthy phenotype. But yet we don't know if it is a good screening parameter, but we can say that shape index could be used as a metabolic screening parameter in obese children and adolescents, but we don't know yet. So we have to study larger samples of obese children to see if this screening parameter has predictive properties to define children with obese children with metabolic complications. So eventually, we could also screen metabolic disorders in childhood obesity with metabolomics. Metabolomics, it's a new tool in the field. So you see, you have genomics, you also following transcriptomics, you have proteomics, and eventually you have metabolomics. In metabolomics, we study the metabolites. Metabolites like fatty acids, lipids, amino acids, sugars, who are present in cell, present in tissue or biofluids like urine, blood, and so on. And we study metabolic responses to various stimuli, so to diseases. We look for a metabolic response of the body or a response on pharmaceutical treatment or genetic modification and so on. At the moment it's used in diagnostics and can be used in diagnostics, drug discovery and development, and also the biotechnology industry. So when we focus on metabolomics, it's very complex. There are lots of metabolites in our body, and they are related with each other, and the pathways are related. So it's a complex tool, but yet it's very interesting to study it. So how do we study it? We have different kinds of how do you say it? different ways to study it. First, we can pre-treat our sample, or we do not. So we can pre-treat it with isotopic labeling or longitudinal sampling. We can process the sample or sample processing of a live organism or biofluids or whole lysate or solvent extractions. We can separate the sample before we study it, or we could not. And we separate it with, for example, gas chromatography, liquid chromatography, and so on. We can detect the signal with nuclear magnetic resonance or mass spectroscopy or spectroscopic radiation. And eventually it could be applied in detection of metabolites, so biomarkers, one or a couple of metabolites. We can also study concentration of fluxes, or we can study it to identify novel compounds. And we can also study the metabolic fingerprint. So in our study, we focused on the sample processing of biofluids, so urine and blood. And we did not do a separation of the sample, but we focused us on nuclear magnetic resonance of protons. And we eventually want to see a metabolic profile or metabolic fingerprint, which is different between obese and non-obese children. So we wanted to compare 75 obese children compared to 75 lean children, but till now we don't have we haven't the number yet, but this is our goal. And we want to identify and compare the metabolic fingerprint of obese and non-obese children using the nuclear magnetic resonance spectroscopy. And we take fasting samples of blood, so we eventually get the plasma from it, and we wanted to study urine samples. I also asked them for personal familial background. I measure them. I measure body fat, length, weight, and blood pressure, and so on. I also asked them to fill out a food diary and physical activity questionnaire, and eventually we can see if this also contributes to the metabolic fingerprint of these children. So we measure it with the magnetic resonance spectroscopy. And we put into the sample, it's putting into the device, and it eventually measures the resonance of the nuclei, the proton nuclei. So we get a whole spectrum, on which the metabolites are. On a specific region of the spectrum, we can detect different metabolites. So if we calculate the integrals, we see in plasma we have 96 integral segments, so area under the curve. For urine, we have less, we have 74 segments, so related with the compound, so the metabolites present in the sample. Eventually, we get a number, and the number of metabolite per patient comes into an Excel data sheet, and then we can compare the control subject with obese, for example. And then we see which metabolites are different, which metabolites are more expressed, or which metabolites are less expressed. And we could also do a discrimination analysis, which means that it's like a statistical method to discriminate groups and to see if there is a difference between metabolic fingerprint or between metabolic profile. Eventually, we can interpret it in biology or biochemical pathways, and we can see which metabolites are up or down regulated in obese persons, and see in which metabolic pathways they are important. So how the process or metabolic metabolic dysregulation in obese children is pronounced. So these are my preliminary results. I detected like it's too low sample size, but I wanted to let you show that the control group, so the lean subjects and obese, were separated very good, meaning that there is a difference in metabolic profile, but we have to see what the difference is and which metabolites are discriminating normal weight and obesity. Okay. That was not my last slide. wanted to let you see that the output, so we see differences in metabolites, for example, glycerol, choline, and so on, and they are related with specific metabolic pathways. So, for example, if glycerol is upregulated in obese children, we see that there would be a dysregulation in lipids, fatty acids, and carbohydrate metabolism, and we can see what the problem is and how obesity may be caused, or what the effect is of obesity. So obesity, induces systemic changes in metabolism, altering a number of biochemical pathways. And we have to implicate the down or upregulation of specific metabolites and through the examination of existing literature. So we have to look into literature and known metabolic pathways to see if some metabolites expressed more or less in obese children are related with known metabolic pathways, and conclude eventually implicated. We also wanted, or in future, I want to correlate these important clinical parameters like BMI, body fat measurements, diet, but also level of physical activity with a certain metabolic profile. So see if diet, physical activity can influence the metabolic profile of obese subjects, for example. And the goal is to identify novel early-stage biomarkers, so biomarkers which can predict obesity and associated health risks like diabetes or cardiovascular disease. So that's the goal, to eventually detect some biomarkers which we can use in the study of obesity in children. So that was my talk. If there are questions, I'd like to answer them. So you may ask if there's something you want to know, or